When you think about carbohydrates, I bet your mind probably jumps straight to energy.
Yeah, pasta, bread, sugar in your coffee.
Exactly. We're sort of trained to see them as just fuel, right?
Yeah.
Calories you count, burn, store.
Mm-hmm.
But if we actually, peel back that diet talk and look at the source material, the chemistry itself,
Yeah.
it's maybe one of the most structurally subtle, mathematically complex, and functionally diverse areas in all of biochemistry. It's kind of amazing.
It really is. It's an area where tiny details, millimeters if you like, at the molecular level, they matter hugely. Historically, people called them hydrates of carbon.
Right, the whole CnH2O formula.
Exactly, because often the empirical formula looked like that, but honestly that definition is chemically pretty insufficient.
It doesn't quite cut it.
No, it's a bit of a relic, really. It doesn't hold up when you look closely.
So if that old definition is inadequate, what's the precise modern chemical rule we should be using?
Well, the sources are really clear on this. Carbohydrates are rigorously defined by their functional groups. They are polyhydroxyaldehydes or polyhydroxyketones or compounds that yield those specific structures when you hydrolyze them, break them down with water.
So the key bits are the multiple hydroxyl groups, the OHs, plus either an aldehyde or a ketone group.
That's it. That combination is the defining feature.
And we really have to use that functional group definition because the old formula just fails for some really crucial molecules, doesn't it?
Precisely. Take D2-deoxyribose, the backbone of DNA.
Can't get much more essential than that.
Right. It's absolutely a carbohydrate derivative central to life, but it doesn't quite fit the CH&H2O formula perfectly because it's missing an oxygen atom. That's why we have to rely on the presence of those critical functional groups.
Okay, so our mission in this deep dive then is to move way past the grocery list view and really understand the secret life of sugar. We want to explore how these incredibly tiny differences in 3D structure, what chemists call stereoisomerism, how they dictate absolutely massive biological outcomes.
Yeah, it's fascinating.
How does just twisting one single bond determine if a molecule fuels your run or builds the shell of a crab or even determines your blood type? It seems incredible.
It's the ultimate lesson in structural humility, you could say. We're showing that life's efficiency relies on absolute specificity. We'll show you why just one misplaced hydroxyl group can make an energy source completely unrecognizable.
And useless to our bodies.
Functionally useless, exactly.
Right. So to really get a grip on this complexity, we need to start with the basics, the scale of these things.
Yeah.
How we categorize them, primarily by size, how many basic units make them up.
We start at the bottom, the foundation, monosaccharides.
Okay.
These are the simplest sugars, the basic building blocks. You can't break them down further into smaller sugar units through hydrolysis.
And these often fit the old formula.
They often do have that generalized H2O matter formula, and importantly, they have all those classic physical properties we think of when we hear sugar.
Meaning, these are the sweet ones.
They're sweet, yes. They're crystalline solids at room temperature, and they're highly water-soluble. Think of glucose. Small, neat, dissolves easily in your bloodstream. That's a typical monosaccharide.
Okay, so moving up the size chart, what's the next category?
That takes us to the oligosaccharides.
Oligo, meaning few.
Exactly. These are molecules that, when you hydrolyze them, they release a modest number of monosaccharide units, somewhere between 2 and 10, usually.
2 down, okay.
Now, while that range is technically correct, the most common and biochemically relevant ones, the ones we'll focus on, are the disaccharides.
Dis meaning 2.
Right. 2 monosaccharide units linked together, like lactose in milk, or maltose from starch breakdown, or sucrose, table sugar.
Got it. And then from the few, we jump straight to the giants, the polysaccharides, or glycans, you call them.
Yes, glycans is another term, polysaccharides. These are the real structural titans of the carbohydrate world.
Big polymers.
Huge, high molecular weight polymers, sometimes built from thousands, even up to 1 million monosaccharide units all linked together. Wow.
And structurally, functionally, they're really the opposite of simple sugars.
How so?
Well, they're generally tasteless. That's why we often call them non-sugars. Think about starch or cellulose. And instead of dissolving clearly in water, like glucose does, they typically interact with water to form sticky, viscous colloids. Think wallpaper paste.
Right. Okay. So once we know we have one of these massive polymers, is there another layer of classification?
Yes, we categorize polysaccharides based on their composition, what they're made of.
Okay.
If they're built from repeating units of only a single type of monosaccharide, say just D-glucose units over and over again. They're called homopolysaccharides.
Homo meaning same.
Exactly. Starch and glycogen are classic examples. They're composed entirely of glucose units, so they're glucans.
And the alternative?
The alternative is heteropolysaccharides, hetero meaning different. These yield a mixture of different sugars or sugar derivatives when you break them down.
And where do we find these? This group includes really crucial components of the body's connective tissue. Things like the mucopolysaccharides, which form the gel-like substance in our joints and skin. We'll definitely talk more about those later.
Okay, let's drill down into the naming system, the grammar, if you like, of those basic monosaccharide units. You said before we even talk 3D shape, we classify them using two key things: the functional group and the number of carbons.
Exactly right. That functional group classification immediately splits all monosaccharides into two main families: aldoses and ketoses.
Aldoses first. What defines them?
An aldose contains an aldehyde group. Chemically, that's a carbonyl group, C double bond O, situated right at the end of the carbon chain, bonded to at least one hydrogen atom.
Can you give us a quick example?
Sure. The simplest possible aldose is glyceraldehyde. That's a 3-carbon sugar, a triose.
Okay.
And probably the most famous aldose, the primary fuel for our brain, is glucose. Glucose is a 6-carbon sugar, a hexose.
Right. And the other side of that coin, the ketoses.
Ketoses contain a keto group. That means the carbonyl group, the C double bond O, is located internally within the carbon chain. It's usually at the C2 position in most biologically relevant sugars.
And examples there.
The simplest ketose is dihydroxyacetone, another 3-carbon triose. But the most prominent ketose in our diet, the one famous for being really sweet, is fructose. That's also a 6-carbon hexose.
Ah, so it's putting these two pieces of information together, the functional group and the carbon count, that gives us the sugar's full, precise name. This is where the naming system, the nomenclature, gets really useful, I imagine.
It's fundamental, absolutely. We use prefixes for the carbon count.
Yeah.
Triose is for 3 carbons, tetrose is for 4, pentose is for 5, hexose is for 6, heptose is for 7, and so on.
So if you tell me glucose is an aldohexose.
You immediately know two things. It's a 6-carbon molecule, hexose, and it carries that reactive aldehyde group at the end of its chain, aldo.
And conversely, fructose is a ketohexose.
Right. 6 carbons again, but this time with the keto group located internally. This chemical language is really the doorway to understanding their potential reactivity and how they behave.
Okay, so we've got the basic classification down. Now, you mentioned earlier that these tiny structural differences are absolutely key. This brings us to the geometry lesson. Stereoisomerism. The idea that you can have two molecules with the exact same list of ingredients, same chemical formula, but because the atoms are arranged differently in 3D space, they're biologically totally distinct.
That's it, exactly.
You used the analogy of keys earlier. Same metal, different cut, fits a different lock.
It's a perfect analogy. Stereoisomerism defines compounds that have the identical structural formula, identical connectivity of atoms, but they differ purely in their spatial configuration. And these differences, they can literally determine whether a molecule is essential food or, just inert material or even toxic. Hashtag TAC 2.1 asymmetry and the isomer formula. Now, the ability of a sugar molecule to even have these different spatial arrangements to form these isomers hinges on one critical feature.
Which is the asymmetric carbon atom. It's often called a chiral center.
And then what makes a carbon asymmetric?
It's a carbon atom that is attached to 4 completely distinct atoms or chemical groups. No two attachments are the same.
Okay. And the number of these asymmetric centers in a molecule tells us something important.
It tells us the potential for complexity. It determines the sheer magnitude of possible structures.
How does that work?
It creates this exponential explosion of possibilities. The number of potential stereoisomers you can have is calculated by a simple formula.
2 to the power of n.
Where n is the number of asymmetric carbons in the molecule. Now let's apply that to our main character, glucose. Glucose has 4 asymmetric carbons. C2, C3, C4, and C5 are all chiral.
Okay, so Nollers is 4. 2 raised to the power of 4. $2 times 22 is 4. $4 times 2 is 8. $8 times 22 is 16. 16. That means there are 16 possible sugars that are all isomers of glucose. That's incredible.
It is incredible. And think about the energy the cell saves by being incredibly specific. Nature had 16 potential ways to arrange those exact same atoms for an aldohexose.
Wow.
Yet life, especially mammalian life, relies almost exclusively on just one of those, D-glucose. And maybe a couple of its very close relatives, like D-galactose and D-mannose, which differ only slightly.
The specificity is just astonishing.
It really shows how crucial that exact geometric fit is for our enzymes, for our entire metabolism.
So out of those 16 theoretical possibilities, we only really encounter a handful inside a human cell. And why D-glucose? Why did life settle on that one as the sort of default fuel rather than, say, DLOs or D-glucose?
We really only see a select few in significant amounts. The choice of D-glucose probably boils down to a combination of factors. Its relative metabolic efficiency, perhaps, and the inherent stability of its preferred cyclic structure, its ring form, which we'll definitely get to.
Okay.
But the absolute key takeaway here is that the cell's machinery, all those enzymes, is sculpted over evolutionary time to recognize the exact shape of D-glucose.
So if you somehow ingested one of the other 15 isomers, for the most part, your enzyme simply wouldn't have the complementary shape. They couldn't bind it properly, they couldn't process it efficiently, if at all. It just wouldn't fit the lock. Hashtag tag 2.2 DL configuration. Enantiomers.
Okay, this brings us to the first major distinction in 3D space. The D versus L configuration. You mentioned these are enantiomers.
Yes, enantiomers. That's the technical term for stereoisomers that are non-superimposable mirror images of each other.
Non-superimposable mirror images. Like your hands.
Exactly like your hands. Your left hand is a mirror image of your right, but you can't perfectly overlay one on top of the other. They're distinct 3D objects. In biochemistry, this DL distinction is absolutely massive.
So how do we assign whether a sugar is D or L? Is there a reference point?
There is. We use the simplest possible carbohydrate that actually has an asymmetric carbon. That's glyceraldehyde, the 3-carbon aldose. That's our reference standard.
Okay. And what's the specific rule for assigning D or L based on that reference? Because I remember reading that the designation doesn't always tell you which way the molecule rotates light.
That is a crucial point. The DL configuration is purely about the structure, the spatial arrangement, not about its optical activity. They don't always correlate.
So what's the rule?
The rule is based on the position of the hydroxyl group, the OH group, on the asymmetric carbon atom that is furthest from the aldehyde or keto group, but adjacent to the terminal primary alcohol carbon.
Okay, that sounds complicated. For glucose, a hexose, which carbon is that?
For hexoses like glucose, that reference carbon is carbon 5, C5. So you look at the standard way of drawing the molecule flat on paper, the Fischer projection. If the OH group on C5 points to the right, it is defined as a D-series sugar.
T for dextro, right?
Well, historically maybe, but structurally just means right in the Fischer projection. If that C5OH group points to the left, it's an L-series sugar.
L for left, okay. And this single structural definition, right versus left on C5, dictates the entire usability of that sugar for us mammals.
Pretty much, yes. What's the biochemical significance of this overwhelming preference for the D-series? It's arguably the single most important rule in carbohydrate metabolism for mammals.
Why?
Because the vast majority, almost exclusively, the naturally occurring monosaccharides found in mammalian tissues are of the D configuration. Predominantly D. This means our entire enzymatic toolkit, thousands of proteins responsible for breaking down sugars, building them up, using them for signals, is stereospecific. It's tailored like a lock to fit only the D-series sugars.
So if I ate L-glucose,
your body would essentially treat it as inert material. It can't be phosphorylated by hexokinase, it can't enter glycolysis, it can't be stored as glycogen. It just doesn't fit the enzymes. That single hydroxyl group orientation makes the difference between essential fuel and, well, just something passing through.
Okay, so D and L forms are complete mirror images. What about epimers? You mentioned them briefly. They sound like a different kind of subtle difference, like a molecule that's almost identical, but off just at one specific point.
That's an excellent way to put it. Epimers are stereoisomers that differ in their configuration around only one specific asymmetric carbon atom.
Just one.
Just one. And importantly, this definition excludes the anomeric carbon, C1 in aldoses, C2 in ketoses, that has its own special designation we'll get to. So think of it as a very localized structural difference compared to the DL mirror image. But that one difference creates an entirely new, distinct molecule.
We have really key examples of sugars that are metabolically essential, but they only differ from our main guy, glucose, by a single OH flip at one carbon.
Absolutely. The classic examples really highlight how a tiny geometric tweak changes everything. Take glucose and galactose.
Okay.
They are identical except for the orientation of the OH group at carbon 4. So they are C4 epimers.
Just C4.
Just C4. Then take glucose and mannose. They are identical except for the orientation of the OH group at carbon 2. So they are C2 epimers.
C2 epimers. And even though they're so similar, they're treated differently by the body.
Yes. Despite sharing the same chemical formula and most of their structure, they often interact with different transport proteins to get into cells, and they might enter slightly different initial metabolic pathways. The body recognizes them as distinct entities.
So if the body needs, say, galactose to make milk sugar, but mostly we absorb glucose from our diet, how does it handle that? Does it have to make galactose from scratch?
This is where the cell shows some flexibility. We have specialized enzymes called epimerases.
Epimeresis makes sense.
Their sole job is to catalyze the interconversion between these epimers. This process is called epimerization.
So they can just flip that one OH group.
Essentially, yes. If your mammary gland cell needs galactose to synthesize lactose and all it has readily available is glucose, an epimerase can flip that C4-hydroxyl group on glucose, turning it into galactose. It's like having a little toolkit to modify the building blocks as needed.
That seems much more efficient than building everything from the ground up.
Exactly. It allows for essential shuffling of these closely related structures without requiring completely separate, complex synthetic pathways for each one.
Now, when we talk about these precise structures, C4 epimers, DL forms, it's really easy to visualize them as these flat, straight chains, like we draw them on paper.
The Fisher projections, yes.
But our sources remind us that in the real biological world, which is mostly water, that linear open-chain form is actually pretty rare, almost irrelevant structurally speaking.
It's very fleeting. It exists, but only transiently as an intermediate. In solution, monosaccharides are much, much more stable when they curl up and form rings.
They react with themselves.
They do. They're reactive molecules. A hydroxyl group on one part of the carbon chain reacts internally with the aldehyde or keto group at the other end. This forms what's called a cyclic hemiacetal if it started as an aldose, or a cyclic hemiacetal if it started as a ketose.
Okay, so taking glucose again as our main example, which specific carbons are involved in this internal chemical handshake to form the ring?
For glucose, it's the aldehyde group at carbon 1, C1, that reaches across the molecule and reacts with the alcohol group, the hydroxyl group, way down at carbon 5, C5.
C1 reacts with C5.
Yes, this closes the chain into a ring. And for glucose, this forms a particularly stable six-membered ring structure that contains one oxygen atom within the ring itself. This six-membered ring structure is called a pyranose ring.
Pyranose for six-member. Is there another common size?
Yes, five-membered rings are also common, especially for ketoses like fructose. A five-membered ring is called a furanose ring. Think pyrin versus furan in organic chemistry. Hashtag 3.1 cyclic structure formation, anomers. Now, arguably the most important consequence of this ring closure, the cyclization, is that it creates a new asymmetric center in the molecule.
A new one.
We're at C1 for aldoses like glucose. Remember, in the open chain form, C1 was the aldehyde carbon. It was double bonded to oxygen and bonded to hydrogen, so it only had 3 different attachments, not 4. It wasn't chiral. But when it forms the hemiacetal in the ring, that C1 carbon becomes bonded to 4 different things. The oxygen from C5, the original hydrogen, the rest of the ring, C2, and a new hydroxyl group formed during the reaction. It's now chiral.
Ah, okay. And this new chiral center has a special name.
It does. We call this new asymmetric center, created by a ring formation, the anomeric carbon.
Anomeric carbon. C1 in glucose?
C1 in glucose, yes. Or C2 in ketoses like fructose. And because this anomeric carbon is now chiral, it can exist in 2 different spatial configurations.
And these are the alpha and beta forms we hear about.
Exactly. These 2 resulting cyclic structures, which differ only in the configuration around that newly formed anomeric carbon, are called anomers.
Alpha and beta anomers.
Yes. And the distinction is purely spatial, how that new OH group on the anomeric carbon is oriented relative to the rest of the ring. In the standard way we draw glucose rings, here with projections, in the alpha anomer, the OH group on C1 typically points down or axially. In the beta anomer, the OH group on C1 typically points up or equatorially. Another way to define it, which works regardless of drawing convention, is relative to the CH2OH group, C6, that sticks out of the ring. In the alpha anomer, the C1OH is on the opposite side of the ring from the C6 group. In the beta anomer, the C1OH is on the same side as the C6 group.
That seems like such a tiny difference. Does it really matter?
It matters immensely because that anomeric hydroxyl group is the one that will participate in forming glycosidic bonds to link sugars together. Whether it's in the alpha or beta position dictates the shape of the resulting bond and the larger molecule, like starch versus cellulose. It's absolutely critical.
Okay, so we have these 2 anomers, alpha and beta. And you said the straight chain form is just a fleeting intermediate. The fact that these forms exist and can sort of flip between each other through that intermediate leads to this phenomenon called mutarotation, right?
Yes, exactly.
This is the actual experimental evidence that sugars aren't just static rings in water, but they're constantly dynamically opening and closing.
Mutarotation is the perfect demonstration of the dynamic nature of sugars in solution. It's defined as the change in specific optical rotation that occurs when you dissolve a sample of a pure anomer, either pure alpha or pure beta, in water.
Optical rotation. That's how the sugar solution bends polarized light.
Precisely. Because stereoisomers interact differently with plain polarized light, the alpha and beta anomers have different initial optical rotation values. But as they sit in solution, they start to interconvert. The rings open briefly to the linear form and then reclose, sometimes as alpha, sometimes as beta.
So the mixture changes over time.
The mixture changes until it reaches a stable equilibrium, a constant ratio of alpha and beta forms. And since the alpha and beta forms rotate light differently, we can actually measure this shift in the overall rotation of the solution as it reaches that equilibrium.
Can you walk us through the actual numbers for D-glucose? That makes it clearer.
Sure. Let's look at the specific data points from the sources. If you take a sample of pure crystalline alpha-2-glucose and dissolve it in water, its initial specific optical rotation is quite strongly positive, plus 112 two-words.
Okay, plus 112.2.
Now, if you take a sample of pure crystalline beta-glucose and dissolve that, its initial rotation is much lower, still positive, but only plus 18.7 to pound.
Big difference. Plus 18.7.
Huge difference. But here's the key. Regardless of whether you started with pure alpha or pure beta, if you let that solution sit, the optical rotation will gradually change over time. It will drift
Until
it stabilizes at a single constant weighted average value, plus $52.07.
always plus 52.7 degrees for glucose.
For D-glucose at equilibrium, yes, that stable value represents the final relaxed state of the system, the equilibrium mixture.
And you said this interconversion happens via that open chain form.
Yes, the ring has to open briefly into the linear aldehyde form, and then it can reclose into either the alpha or beta ring. But that linear form is really insignificant concentration-wise. Maybe only 1% or less of the molecules are in that form at any given instant.
Right. And at equilibrium, is it a 50-50 mix of alpha and beta?
No, it's not. The beta anomer is actually slightly more stable, thermodynamically favored, in aqueous solution. Part of the reason is that its bulky OH group on C1 is often in the more spacious equatorial position in the chair conformation, leading to less steric hindrance.
Okay, so beta's more stable. What's the ratio then?
So at equilibrium, the beta anomer predominates. The final mixture is about 63% beta-glucose and about 36% alpha-D-glucose, with that tiny trace of the open chain form making up the rest. The plus 52.7 degrees is the weighted average rotation of that specific mixture.
Fascinating. And we see the same dynamic opening and closing, this mutarotation, in ketoses like fructose too. You mentioned fructose can form different ring sizes.
Absolutely. Fructose also undergoes mutarotation, and it's structurally even more flexible, you could say. While glucose strongly prefers that six-membered pyranose ring, fructose readily interconverts between the pyranose, six-membered, and the furanose, five-membered ring forms in solution.
So it's even more dynamic.
In a way, yes. This conformational flexibility, being able to adopt different ring shapes, is thought to be one reason why fructose tastes significantly sweeter than glucose. Different receptors in our taste buds might recognize and bind to these different forms. When fructose reaches its equilibrium in solution, its final rotation stabilizes at 92 testolar.
Minus 92, so it rotates light strongly to the left.
Yes, fructose is strongly levorotatory, hence its other name, levulose. Glucose, being dextrorotatory, is also called dextrose. Hashtag, 3.3, reducing properties and interconversion.
Okay, so that anomeric carbon, C1 in glucose, the one that's formed during ring closure and is involved in mutarotation,
Yeah.
that's also the key to another major property of sugars, their ability to act as reducing agents.
Exactly. That anomeric carbon, specifically its ability to revert back to the open chain form with its free aldehyde or keto group, is what gives many frizzlers their chemical superpower, as you called it.
The power to reduce things. Why is that important?
It's historically been vital for detecting and quantifying sugars, like in the Benedict's test for glucose in urine. The property is especially pronounced when the sugar is in an alkaline or basic solution.
What happens in alkaline conditions?
Under alkaline conditions, sugars undergo a critical structural rearrangement called tautomerization. It involves the migration of a hydrogen atom and a shift in the position of a double bond.
Okay, tautomerization. What does that produce?
This process generates highly reactive intermediate structures called enedials. Enia for the double bond, diol for two hydroxyl groups attached to the double bonded carbons.
Enediols, and these are the reactive species.
Yes, these enedials are unstable and very readily oxidized. Because they get oxidized so easily, they can act as potent reducing agents. They donate electrons to other molecules, like the copper ions in Benedict's region, reducing the copper from blue C2+ to reddish Q+ Le.
And this ability is directly linked back to that anomeric carbon being able to open up.
Directly. If the anomeric carbon is locked, say in a glycosidic bond, like in sucrose, the ring can't open, no enediol can form, and the sugar is non-reducing. So we call sugars with a free or potentially free aldehyde keto group reducing sugars.
Now, this reactive enediol intermediate, that's also the explanation for something quite surprising. The fact that three major sugars, glucose, fructose, and mannose, can actually turn into each other under certain lab conditions.
Yes, that's the Lobry-de Broglie-von-Eckenstein transformation. It sounds complicated, but the core idea relies on that shared enediol intermediate.
How does that work? How does a shared intermediate bridge the structural gaps between glucose, fructose, a ketose, and mannose, a C2 epimer of glucose?
Okay, think about their structures near the top. Glucose and mannose differ only at C2. Fructose is a ketose, so its structure is different at both C1 and C2 compared to glucose. However, when any of these three sugars are placed in mild alkaline conditions, they all tautotomerize to form the same 1-Gov2 enediol intermediate.
They all converge on the same intermediate structure.
For that C1-C2 region, yes. The double bond forms between C1 and C2, and the stereochemistry at C2 is temporarily lost. Since they all share this common intermediate structure, the process is reversible. The enediol can collapse back down, reforming the carbonyl group, but it can resolve into any of the three starting sugars.
So you can start with pure glucose and alkali and end up with a mixture of glucose, fructose, and mannose.
Exactly. It's an equilibrium process. This transformation really underscores just how dynamic and somewhat fluid the structural distinctions between these closely related sugars can be under specific chemical conditions.
And that explains the really interesting results you get with a specific chemical test called osazone formation.
It connects directly to that, yes. The osazone test involves reacting a sugar with an excess of a reagent called phenylhydrazine. This reaction specifically involves carbons 1 and 2 of the sugar.
C1 and C2 again.
Right. Since the structural differences between glucose, fructose, and mannose only exist around C1 and C2, and since these carbons become heavily modified and essentially masked by reacting with 3 molecules of phenylhydrazine,
The final product ends up being the same for all three.
precisely, glucose, fructose, and mannose all yield the exact same osazone crystals, which are characteristically needle-shaped. This is strong chemical evidence that their structural differences are localized to that C1-C2 region, the same region involved in the enediol interconversion. Galactose, which differs at C4, forms a different shaped osazone.
Okay, we've really dug into the complexity of a single sugar unit, the isomers, the rings, the anomers, the reactivity. Now let's move to linking them together. Polymerization. When two or more monosaccharides join up, they form what's called a glycosidic bond. And the formation of this bond seems crucial because it completely locks in that alpha or beta configuration at the anomeric carbon that we just spent time detailing.
Absolutely. The glycosidic bond is the fundamental covalent linkage that builds up all the complex carbohydrates, from simple disaccharides to massive polysaccharides like starch and cellulose.
How does it form?
It typically forms when the hydroxyl group on the anomeric carbon, C1 or C2 of one sugar, reacts with any hydroxyl group on another molecule.
It doesn't have to be another sugar.
No, often it is, linking sugar to sugar. But that second molecule could also be a non-carbohydrate component, like an alcohol, a phenol, or even a nitrogen atom in an amine. If the second part isn't a sugar, we call it an aglycone. The whole molecule is then called a glycoside. Hashtag tag 4.1, the glycosidic bond and nomenclature. But the absolutely critical point, getting back to anomers, is the specific stereochemistry, whether the reacting anomeric carbon was in the alpha or beta Collocker configuration before the bond formed. Because once that bond forms, the ring is locked. It can no longer open and close at that position.
So that alpha or beta designation becomes permanent in the linkage.
It becomes a permanent structural feature of the bond itself. And this dictates everything, the shape of the resulting molecule, which enzymes can recognize and break that bond, etc.
So when we see chemists or biologists write out a linkage like, say, beta 1 right RO4R, that notation isn't just shorthand. It's a precise structural instruction manual.
It tells you everything you need to know about that specific connection. The nomenclature of the glycosidic bond is incredibly informative. Let's break down the beta-right RO4 or.
Yep.
The clay printout tells you it involves the anomeric carbon C1 of the first sugar. The 4 tells you it connects to the hydroxyl group on C4 of the second sugar. And the beta tells you the crucial stereochemistry. The anomeric carbon of that first sugar was in the beta configuration when the bond formed.
So just 3 symbols tell you the connection points and the 3D orientation.
Exactly. An alpha-1 right RO4 bond, like in starch, is fundamentally different geometrically from a beta-right RO4 bond, like in cellulose.
And this structural lock, formed by the glycosidic bond, is also what determines whether the resulting disaccharide is classified as reducing or non-reducing, which you mentioned earlier.
Correct. It depends on which hydroxyl groups are involved. If a disaccharide is formed such that one of the original monosaccharide units still has a free anomeric carbon, meaning its ring can still open up to potentially expose an aldehyde or keto group, then the whole disaccharide remains a reducing sugar. Maltose and lactose are examples.
Okay.
But if the glycosidic bond is formed between the anomeric carbons of both sugar units, then neither ring can open up. Both potentially reducing groups are locked in the bond. In that case, the molecule becomes a non-reducing sugar. Sucrose is the classic example. It can't form anidiols, can't reduce copper ions. Tagged 4.2 biochemically important disaccharides.
Right, let's quickly review the 3 giants of the disaccharide world, highlighting these features, starting with maltose or malt sugar. We'll get this from breaking down starch, right?
Yes, maltose is essentially 2 glucose units stuck together. Specifically, it's 2 alpha-D glucose units.
And the linkage.
They are linked by an alpha-1 right RO4 glycosidic bond. So the anomeric C1 of the first glucose in its alpha form connects to the C4 hydroxyl of the second glucose.
Okay, alpha-1 rhido 4, and is it reducing?
Yes, because the anomeric carbon C1 of the second glucose unit is not involved in the bond. It's free. So that ring can still open and close, meaning maltose is indeed a reducing sugar.
When if you did the ozazone test on it.
Maltose yields characteristic, beautiful sunflower-shaped ozazone crystals, different from the needle shape of glucose-fructose mannose.
Okay, next up, lactose, the famous milk sugar critical for babies.
Lactose is structurally distinct. It's composed of a beta-D galactose unit linked to a D-glucose unit. Remember, galactose is the C4 epimer of glucose.
Right, and the linkage here.
The bond is a beta-1 right RO4 glycosidic bond. So the anomeric C1 of the galactose in its beta form connects to the C4 hydroxyl of the glucose.
Beta 1,4 this time. Reducing or non-reducing?
Still reducing. Again, the anomeric C1 of the glucose unit is free and available to open up.
Makes sense.
And the sources really emphasize its fundamental importance in nutrition, noting it's the most crucial carbohydrate source for young mammals. If you test lactose, it yields powder puff, or sometimes described as hedgehog-shaped ozazones.
Powder puff. Okay, and finally, sucrose. Ordinary table sugar. You said this one is the structural anomaly in the family.
Sucrose is quite unique because it links an aldose, glucose, with a ketose, fructose. Specifically, it's formed from alpha-D-glucose and beta-D-fructose.
Alpha-glucose and beta-fructose, how are they linked?
The linkage is highly specific. It's an alpha-1 right arrow beta-2,2 glycosidic bond. This means the anomeric carbon of glucose, C1 in alpha form, is linked directly to the anomeric carbon of fructose, C2 in beta form.
Ah, so it links anomeric carbon to anomeric carbon?
Exactly. The potentially reducing groups of both sugar units are tied up, locked into that glycosidic bond.
And since both reducing sites are locked, the molecule has no way to open up, no way to form an anediol.
Correct. Sucrose is therefore a completely non-reducing sugar, which also means diagnostically it cannot form ozazones. That's a key chemical difference setting it apart from maltose and lactose.
And this unique lock structure is also directly responsible for that phenomenon you mentioned earlier called inversion.
Yeah.
It sounded complicated, but you said it's really just a trick of light rotation upon breaking the bond.
It is. It's a rather striking laboratory observation, though. Pure sucrose in solution is dextrorotatory. It rotates plain polarized light to the right. The specific rotation is plus $66.05.
Okay, positive rotation.
But when you hydrolyze sucrose, break that alpha-1 right arrow beta-2,2 bond using either acid or a specific enzyme called invertase or sucrase, you release a 1-to-1 mixture of free glucose and free fructose.
Right.
Now, we know glucose at equilibrium is dextrorotatory, plus $52.07. But fructose, as we established, is strongly levorotatory, 92 test.
Ah, so the negative rotation of fructose is much stronger than the positive rotation of glucose.
Exactly. The strong negative rotation of the liberated fructose dominates the mixture. The resulting one-to-one mix of glucose and fructose, called invert sugar, has an overall negative specific rotation around $28.02.
So the rotation flipped from positive to negative.
It inverted, hence the name inversion for the hydrolysis process and invert sugar for the resulting mixture. It's just a consequence of the optical properties of the constituent monomers versus the original disaccharide.
Okay, great. We've covered the dimers. Now let's shift focus fully to the really big polymers, polysaccharides. We're moving from these relatively small, often soluble sweet structures to these huge, often dense molecules that serve, as you said earlier, two main kinds of roles, either massive energy reserves or physical scaffolding and fortification.
That's a perfect summary of their major functions, yes. Hashtag tag 5.1 homopolysaccharides, storage and fuel. Let's start with the storage ones, built from a single type of monomer, homopolysaccharides. And the absolute king here, certainly in plants and in our diet, is starch.
The most important carbohydrate we eat, basically.
Arguably, yes. It's the primary way plants store energy. Structurally, it's a polymer built entirely of D-glucose units, making it a glucan. But starch isn't actually one single substance. It's typically a mixture of two different glucan structures.
Two components in starch. What are they?
There's amylose, which is essentially a long, linear, unbranched chain of glucose units linked purely by alpha-1 right arrow 4 glycosidic bonds. This structure tends to coil up into a helix.
Okay, amylose is the straight chain.
Mostly straight chain, yes. The other component is amylopectin. This also has a backbone of alpha-1 right arrow 4 linked glucose units, but it's highly branched.
Branched how?
At various points along the alpha-1 right arrow 4 chain, additional short chains branch off, connected via alpha-1 right arrow 6 glycosidic bonds. So amylopectin is this big, bushy molecule compared to the linear amylose. Most starches are predominantly amylopectin.
Got it. And what about the equivalent energy storage molecule in us, in animals?
That would be glycogen. It's often referred to as animal starch because its function is analogous. It's our readily accessible glucose reserve.
Where do we store it?
Primarily in the liver, where it helps maintain blood glucose levels, and in muscle tissue, where it provides fuel for muscle contraction.
And structurally, how does glycogen compare to starch?
Structurally, glycogen is very similar to amylopectin. It's also a branched glucan with alpha-1 right arrow 4 more backbones and alpha-1 right arrow 6 level branch points. The main difference is that glycogen is even more highly branched than amylopectin.
More branches. Why is that important?
That high degree of branching is functionally crucial. It creates many, many non-reducing ends on the molecule. Enzymes that break down glycogen, like glycogen phosphorylase, work from these ends. So having lots of branches means lots of ends for enzymes to attack simultaneously.
Which means faster glucose release.
Much faster mobilization of glucose units to meet sudden immediate energy demands, like during exercise or stress. It's a very efficient structure for rapid fuel delivery.
Okay. Now, we have to briefly mention inulin here. You brought it up earlier. It's a homopolysaccharide, but its importance isn't as fuel, but rather as a diagnostic tool.
That's right. Inulin is unique among the storage polysaccharides we usually discuss because it's a polymer of fructose, not glucose. So it's a fructosan or fructan.
A chain of fructose units.
Yes. And the key thing is, the human body lacks the enzymes needed to hydrolyze the specific glycosidic bonds in inulin. We can't digest it, we can't metabolize it.
So it just passes through us.
Essentially, yes.
Yeah.
It gets filtered by the kidneys but isn't reabsorbed or secreted by the renal tubules. This inertness, this predictable filtration, makes it incredibly useful clinically for assessing kidney function. Doctors can inject a known amount of inulin into the bloodstream and then measure how quickly it appears in the urine and its concentration. Because it's cleared solely by glomerular filtration, this measurement allows for a very precise determination of the glomerular filtration rate, or GFR, a key indicator of overall kidney health. Hashtag tag 5.2 homopolysaccharides, structural roles.
Okay, we've covered energy storage. Now let's switch to the homopolysaccharides that provide rigid physical structure. You mentioned cellulose in plants and chitin in invertebrates earlier, and you hinted that the structural difference between soft starch and hardwood is incredibly tiny chemically.
It's probably the most dramatic illustration in all of biochemistry of how a subtle difference in stereochemistry dictates a massive difference in physical properties and biological function. Let's look at cellulose.
The main component of plant cell walls, right? Wood, cotton fiber.
Exactly. It's the most abundant organic polymer on Earth. Structurally, like starch, it is also a polymer of D-glucose units, a glucan.
Okay, so far sounds the same as starch. What's the difference?
The only chemical difference lies in the glycosidic linkage between the glucose units. Instead of the alpha-1 right-or-out link found in starch and glycogen, cellulose utilizes the beta-arrow glycosidic bond.
Wait, that's it. The only difference between digestible helical starch and rigid indigestible wood fiber is the orientation of that bond at C1, alpha versus beta.
That is the entire difference at the chemical linkage level. It's almost unbelievable, but it's true.
How does that one change cause such a massive difference in properties?
The alpha linkage in starch allows the polymer chain to adopt a flexible, coiled, helical conformation. This structure is relatively accessible to water and, crucially, to the digestive enzymes, amylases, in our saliva and gut that are designed to break alpha-1 right-or-4 gall bonds.
Okay. And the beta linkage?
The beta linkage in cellulose forces the glucose units into an extended linear chain conformation. These straight chains then align parallel to each other, forming extensive networks of hydrogen bonds between adjacent chains.
So they pack together really tightly.
Incredibly tightly, forming strong, rigid microfibrils. This structure provides immense tensile strength, perfect for a plant cell wall, but it's also very resistant to chemical attack. And critically, mammals, including humans, completely lack the necessary enzyme called cellulase that can specifically cleave that tough beta-right-or-forward glycosidic bond.
So we simply can't digest it. It's inedible for us.
Nutritionally, yes, we get no calories from cellulose.
But despite being indigestible, our sources still highlight it as a crucial part of a healthy diet. Why?
Because it forms the bulk of what we call dietary fiber. While it provides no energy, it plays essential physiological roles in the digestive tract.
Such as?
Fiber adds bulk to the intestinal contents, which helps stimulate peristalsis, the muscle contractions that move food along. It can also slow down the rate at which glucose from digestible carbohydrates is absorbed, helping to moderate blood sugar spikes after a meal. Some fibers can bind cholesterol and bile acids, aiding their excretion, so it's highly functional for gut health, even though it's not strictly nutritional.
Functional without being fuel.
Yeah.
Got it. And the other major structural homopolysaccharide you mentioned was chitin. What's its role?
Chitin serves a very similar structural purpose, but primarily in the animal and fungal kingdoms. It's the main structural polysaccharide found in the tough, protective exoskeleton of invertebrates. Think of the shells of insects, crabs, lobsters. It's also found in the cell walls of fungi.
So it's like the cellulose of the invertebrate world. What's it made of?
It's structurally very similar to cellulose. It's also a long, unbranched polymer linked by beta-1 right-RO4 glycosidic bonds. The difference is the monomer unit. Instead of glucose, chitin is a polymer of N-acetyl D-glucosamine, which is essentially glucose with an acetylated amino group attached at C2. That modification doesn't change the overall structural principle of forming rigid fibers via beta linkages. Hashtag 5.3 heteropolysaccharides, the tissue matrix.
Okay, we've covered the rigid structures made from single sugar types. Now let's talk about biological materials that need to be more flexible, maybe elastic or shock absorbent or lubricating. This brings us to the other class of polysaccharides, the heteropolysaccharides, the ones made from different repeating units. And the major group here seems to be the glycosaminoglycans, or GAGs.
Yes, glycosaminoglycans, or GAGs, are absolutely fundamental to the structure and function of our connective tissues. They're also historically called mucopolysaccharides because they often form viscous, mucus-like solutions.
And these aren't about rigidity like cellulose.
No, their role is quite different. They typically form the essential hydrated gel-like ground substance or extracellular matrix, ECM. This is the stuff that fills the spaces between cells in tissues like cartilage, skin, tendons, and blood vessels. It's the material within which the fibrous proteins like collagen and elastin are embedded.
So they provide the supportive, squishy environment. What are they made of?
They are typically long, unbranched chains composed of repeating disaccharide units. And crucially, these repeating units almost always contain an amino sugar, like N-acetylglucosamine or N-acetylgalactosamine, and often an acidic sugar, like glucuronic acid. Many GAGs are also heavily sulfated, meaning they carry sulfate groups.
Amino sugars, acidic sugars, sulfates. Sounds like they carry a lot of negative charge.
They do. They're highly negatively charged polymers. This high charge density causes them to repel each other and attract large amounts of water, forming that hydrated space-filling gel which resists compression, perfect for rolls in joints and cartilage.
Right. Let's look at some key GAG examples from the sources that perform these really specialized non-fuel roles, starting with hyaluronic acid.
Hyaluronic acid, or hyaluronan, is a bit unique among GAGs as it's non-sulfated and incredibly large. It's a major component of synovial fluid, the fluid that lubricates our joints. Its high viscosity makes it an excellent lubricant and shock absorber, cushioning bones during movement.
Okay. Joint lubrication, any other roles?
Yes, fascinatingly, it plays a completely different role in reproduction. It forms a thick, gel-like matrix around the ovum, the egg cell. For fertilization to occur, sperm must penetrate this barrier. They do so by releasing an enzyme called hyaluronidase, which specifically breaks down hyaluronic acid.
Wow, so it's both a mechanical cushion and a kind of reproductive gatekeeper, versatile molecule.
Incredibly so.
Okay, what about some other key GAGs? You mentioned some have roles in blood or tissue support.
Yes. Heparin is probably the most well-known JAG clinically. It's produced by mast cells and is found lining blood vessels. Its major function is as a potent natural anticoagulant. It strongly inhibits blood clotting, which is why it's widely used medically to prevent or treat blood clots.
An anticoagulant built from sugars.
Okay.
And for tissue shape and structure.
Then we have the chondroitin sulfates. These are major components of cartilage, bone, skin, tendons, and ligaments. They are crucial for maintaining the structural integrity and shape of these tissues, largely due to their ability to bind water and resist compressive forces. They literally help give tissues their resilience and form.
Makes sense for cartilage especially. And finally, the sources mentioned one critical for vision.
That's keratin sulfate. This JAG is particularly important in the structure of the cornea, the transparent outer layer of the eye. It also contributes to bone and cartilage structure. Its specific arrangement and hydration properties within the corneal tissue are essential for maintaining the transparency of the cornea.
Transparency relies on a sugar structure.
Absolutely. If the structure or amount of keratin sulfate is abnormal, as happens in some genetic conditions, the cornea can become cloudy, severely impairing vision. It really drives home the point. These complex carbohydrates are essential for life's most intricate functions, far, far beyond just providing energy.
All right, we're moving now beyond the big structural and storage roles into even more specialized functions of carbohydrates and their derivatives, how they interact with our genetics, our immune system, and other really specific physiological processes. Hashtag tag 6.1 genetic structure and metabolic intermediates.
Well, you can't get much more specialized or fundamental than the building blocks of our genetic material. The 5-carbon sugar D-ribose is an absolutely critical component of RNA, ribonucleic acid, the molecule involved in translating genetic information into proteins.
Ribose and RNA.
Okay, but it doesn't stop there. Ribose is also the core sugar component of several essential nucleotide coenzymes. When we talk about the cell's energy currency, we mean ATP, adenosine triphosphate. The A involves adenine, but the TP part is linked via ribose.
So the energy carrier itself is built on a sugar.
Yes. And the same goes for the crucial electron carriers in metabolism. NAD+, nicotinamide adenine dinucleotide, and NADP+, its phosphate version. Ribose forms the structural backbone connecting the nicotinamide and adenine parts in both of these essential redox cofactors.
Wow, so ribose is central to RNA, ATP, NAD plus NADP.
Yeah.
Pretty vital stuff.
Absolutely fundamental.
And the sugar in DNA is just a slight modification of ribose, right?
Exactly. The structural constituent of DNA, deoxyribonucleic acid, is D2-deoxyribose. As the name implies, it's simply ribose that is missing the oxygen atom at the C2 position.
Deoxy. Got it.
That single oxygen atom difference is profound. It contributes to the greater stability of DNA compared to RNA, making DNA better suited for long-term storage of genetic information, while RNA handles the more transient information transfer and catalytic roles. Again, a tiny structural change dictates a massive functional divergence.
It's a recurring theme. And even within the active metabolic pathways themselves, like when we're breaking down glucose, sugars exist as intermediate forms, not just the start and end points.
Constantly. Monosaccharides and their phosphorylated derivatives are the bread and butter of central metabolism. In glycolysis, the main pathway for glucose breakdown, we see crucial intermediates like glucose 6-phosphate, fructose 6-phosphate, fructose 1006-bisphosphate, and glyceraldehyde 3-phosphate. These aren't just fleeting steps, they are key regulatory points and branch points to other pathways.
And other pathways too.
Yes, for instance, the hexose monophosphate shunt or pentose phosphate pathway, which is crucial for generating NADPH, needed for biosynthesis and combating oxidative stress, and producing ribose for nucleotides. That pathway involves specialized sugar phosphates like D-ribulose 5-phosphate and D-cetoheptulose 7-phosphate, a 7-carbon sugar. These intermediates are the chemical currency being passed along the metabolic assembly lines. Hashtag 6.2, cellular recognition and immunology, glycoconjugates. So if the inside of the cell is using sugars primarily for energy and as metabolic building blocks, the outside of the cell often uses them for communication and recognition.
How does that work?
This happens when carbohydrates are covalently attached to other biomolecules, primarily proteins or lipids, forming structures called glycoconjugates. If a carbohydrate chain, lichen, is attached to a protein, it's a glycoprotein. If it's attached to a lipid, it's a glycolipid.
And these glycoconjugates are often found where?
They are major components of the outer surface of cell membranes, with the carbohydrate portions typically extending outwards from the cell surface into the extracellular environment.
So they act like little flags or antennas on the cell surface.
That's a great analogy. These carbohydrate chains mediate a huge range of crucial cellular interactions. They're involved in cell growth regulation, how cells adhere to each other to form tissues, cell adhesion, and specific recognition events like how sperm recognizes an egg during fertilization.
And they're important in the immune system, too.
Hugely important. Many key proteins involved in immunity are glycoproteins. Think of immunoglobulins, which are antibodies, the Y-shaped proteins that recognize and neutralize pathogens. The carbohydrate portions on antibodies are critical for their stability and function. Other glycoproteins like fibronectin and laminin, which are components of the extracellular matrix, are vital for cell recognition, adhesion, and guiding cell migration during development and wound healing. They help define tissue boundaries and maintain tissue integrity.
Okay. But probably the most famous and easily understood example of carbohydrates defining our identity is through blood groups, right?
Absolutely. The ABO blood group substances, the antigens found on the surface of your red blood cells that determine whether you are type A, B, AB, or O, are classic examples of glycoconjugates. They can be other glycoproteins or glycolipids.
And the difference between A, B, and O is purely down to the sugar part.
Purely down to the specific structure of the short carbohydrate chain, oligosaccharide, that's attached. All blood types share a common core oligosaccharide structure. Type O individuals have just that core structure. Type A individuals have an enzyme that adds an extra N-acetylgalactosamine sugar to that core. Type B individuals have a slightly different enzyme that adds an extra galactose sugar instead. Type AB individuals have both enzymes, so they add both sugars.
So a single sugar difference determines your blood type and who you can safely receive blood from.
Exactly. It's the ultimate example of how a very specific carbohydrate structure dictates individual immunological identity and has major clinical consequences. Hashtag 6.3, unusual in clinical functions.
We're almost at the end. But the sources highlight a few more fascinating, sometimes unusual, and clinically relevant roles for sugars and their derivatives. Things that seem completely removed from basic energy or structure.
Yes, there are some really specialized cases. Let's start with the darker side. Pathology linked to sugar derivatives.
Okay.
In certain conditions, particularly uncontrolled diabetes where blood glucose is consistently very high, some cells start shunting excess glucose into alternative metabolic pathways. One such pathway converts glucose into a sugar alcohol called sorbitol and galactose into dulcidol.
Sugar alcohols, what's the problem with them?
The problem is that these sugar alcohols are not easily metabolized further in some tissues, like the lens of the eye, nerves, or kidneys. They tend to accumulate inside the cells. And because they are osmotically active molecules...
Pump no water into the cells.
Exactly. They draw water in by osmosis, causing the cells to swell and eventually sustain damage or dysfunction. This osmotic stress is believed to be a major contributing factor to some serious diabetic complications, including cataract formation in the lens, peripheral neuropathy, nerve damage, and nephropathy, kidney damage.
Wow, so sugar accumulation causing physical damage through osmosis. Okay. Now, on the flip side, on the therapeutic side, the sources mentioned a synthetic sugar being used to treat a specific liver-related disorder.
Yes, that's lactulose. It's a synthetic disaccharide made from galactose and fructose, linked in a way that human intestinal enzymes cannot break down.
So, like inulin and cellulose, it's not absorbable for us.
Correct. Because it's not absorbed in the small intestine, it travels intact to the large intestine, the colon. There, resident bacteria can ferment it.
And what does that achieve?
Lactulose is used primarily to treat hepatic encephalopathy. This is a serious neurological condition caused by advanced liver failure, where the liver can no longer effectively detoxify ammonia produced in the gut. High ammonia levels in the blood are toxic to the brain.
So how does lactulose help?
When bacteria in the colon ferment lactulose, they produce acidic byproducts like lactic acid and acetic acid. These acids lower the pH of the colon. In this more acidic environment, the toxic ammonia readily picks up a proton, H+, and gets converted into the ammonium ion, and H3 becomes NH4 plus SAR.
NH3 becomes NH4 plus mighty. Why is that better?
Because the ammonium ion, sex in H4 plus an Iris charge, it is much less readily absorbed across the intestinal wall back into the bloodstream compared to the uncharged ammonia. So the ammonia gets trapped in the colon as ammonium and is then simply excreted in the feces. It's a clever way to lower toxic blood ammonia levels using a non-absorbable sugar.
That is clever. Trapping the toxin using acidity generated from sugar fermentation. Okay, finally, let's just quickly revisit those two really cool examples that highlight the extreme versatility of sugar chemistry, one from nature, one from medicine.
Right. First, the sheer evolutionary ingenuity seen in certain Antarctic fish. They survive in seawater that's actually below the normal freezing point of water. How? They produce specialized antifreeze glycoproteins.
Antifreeze made of sugar and protein. How does it work?
These molecules don't lower the freezing point significantly like salt does. Instead, they work by adsorbing or binding onto the surface of any tiny ice crystals that start to form in the fish's body fluids. By binding to the crystal lattice, they physically prevent the crystals from growing larger. It's a non-colligative mechanism, inhibiting ice growth rather than lowering the freezing point overall. It's carbohydrate chemistry acting as nature's sophisticated survival tool.
Amazing. And the second example was antibiotics.
Yes. Many important antibiotics are actually glycosides, meaning they have a sugar component attached to a non-sugar aglycogen part. Our sources specifically cite streptomycin as a classic example. Streptomycin is an aminoglycoside antibiotic, effective against certain types of bacteria. The complex carbohydrate portion is absolutely integral to its mechanism of action, which involves interfering with bacterial protein synthesis. It shows that complex sugars can be powerful weapons in the fight against infection. Hashtag outro.
So where does this leave us? We started this deep dive thinking about carbohydrates as, simple fuel, counting calories from bread or sugar. And we end up having seen them as the architects of wood and cartilage, the communication flags on our cell surfaces, the literal backbones of our genetic material, and even as molecular antifreeze or life-saving drugs. It's quite a journey.
It really is. I think the biggest takeaway is that life, at its most fundamental level, is built upon the most precise, intricate molecular geometry. We've navigated almost the entire carbohydrate spectrum today. From that basic DL distinction that determines if a sugar is even useful to us metabolically, to the subtle difference between an alpha and a beta linkage at C1 that dictates whether a glucose polymer becomes digestible fuel, like starch, or indigestible structural fiber, like cellulose.
Every single role seemed to hinge on knowing exactly where each atom, especially those hydroxyl groups, was pointing in 3D space.
Exactly. Think about the lubricant function of hyaluronic acid in your joints, or the absolute transparency required of keratin sulfate in your cornea. These highly specialized functions all depend utterly on the precise conformation and chemical properties derived from that specific sugar structure.
And that profound specificity brings us right back to where we started, really. That almost absolute preference our bodies show for D sugars over L sugars.
It does. We emphasize that our entire complex enzyme machinery built up over millions of years of evolution is exclusively dedicated to recognizing and processing D sugars.
It's an absolute uncompromising preference.
It really is. And maybe the final thought to leave you with is just to ponder the sheer efficiency and perhaps vulnerability of that. Imagine if you could magically synthesize a batch of L-glucose, the perfect mirror image of our normal fuel, and you consumed it.
My body would just ignore it.
It would be functionally invisible to your metabolic pathways. The most basic, fundamental energy pathway that powered your brain, your muscles, the pathway that arguably enabled all complex life to evolve. It hinges entirely on the specific spatial orientation of one single hydroxyl group on one specific carbon atom, C5. This simple yet profound geometrical law is truly the secret life of sugar.